So welcome. I think maybe we can do a fast round just to introduce ourselves. I'm Gustavo Dziewczapolski, scientific director at CMD. I'm not one of the experts. The experts are this side. But my first contact with the CMDs was through LMNA. The first focus meeting I participated in, that was part of this series that is culminating with this big conference, was the LMNA meeting, the third workshop meeting in Paris in October. So.
Hi everyone, I am Anne Bertrand, I am a scientist. I'm working in Dziewczapolski's lab since 10 years, a bit more than 10 years. So I am doing basic research to try to understand what LMNA function is and what is not working with mutations. And we are also working on gene therapy on a mouse model and cellular model to try to find a cure for the disease.
Giselle-Bon's group is the group that found the first mutation related to CMD in 1999.
Okay, hello, I am Susana Quijano-Roy. I come from Spain, but in fact, I work in Paris for more than 17 years now. And I went into layman world because we had a number of patients who had no diagnosis, and at a given moment we realized that they looked very much. Among them, we thought it could be the same disease, and somehow we ended finding that they were layman AC mutated patients. Since that moment, I think it was in 2007-8, I've seen many patients. They are very recognizable, and we have started an effort to try to understand better also how they evolve, because this is a very important thing for families to know how can we help and prevent some of the complications. And well, I'm clinician, so I always learn of patients and families and also the other colleagues. So this is why I'm here. So I would like to...
Hello, I am Soledad Monjes. I'm from Buenos Aires, Argentina. I'm pediatric neurologist, and I have some patients with LMNA, so I'm glad to be here.
My name is Christine. This is my husband, Jason. We have 1.5-year-old named Christian who has Lamin-related congenital muscular dystrophy.
I'm Dr. Felice Heller. I'm a pediatric cardiologist from Connecticut Children's Medical Center, and I have taken care of several children with LMNA muscular dystrophy and collaborated with Susana and several other authors to publish a case report.
Hello, my name is Harris Diggs. My wife Stephanie, we're from upstate New York, Albany. We have a son, 9-year-old Charlie, who suffers from LMNA.
Hi, I'm Allison Ballard. I'm a nurse practitioner at the Multidisciplinary Neuromuscular Clinic in Denver, and I provide patients and families kind of a point of contact and to all the different multidisciplinary care.
Hello, I'm Kyle. My wife Melissa, we're from North Dakota. We have a 3.5-year-old son who has LCMD.
Hello, I'm Shaminder Connersman. I'm a neuromuscular specialist at Rady Children's Hospital and UCSD, and I take care of several patients, adult and pediatric, with LMNA mutations.
Hi, I'm Jenny Vesnal. I'm Director of Research at Muscular Dystrophy in the United Kingdom, and we obviously have families with LMNA mutation.
Hi, I'm Pomi. I'm a student, hopefully going to medical school one day. But for now, I'm working with this international cohort. Also, Susana's involved, and Giselle, Dr. Bonham, and Dr. Muntoni from London to put an international cohort together to sort of iron out the details on what is all involved in children who do suffer from this, so that we can sort of prepare for other patients along the way.
Hi, I'm Sabrina Yum. I'm a neuromuscular specialist at Children's Hospital of Philadelphia. We do take care of a large number of neuromuscular patients, including patients with LMNA mutations, and work very closely with Dr. Barneman, who actually was at CHOP before, on the undiagnosed disease group.
Hi, I'm Jody Westbrook. I'm a scientist at Invitae, and we're a genetic diagnostics lab, and we do panel testing for neuromuscular diseases, including this one.
He's shy. I'm Betsy. This is my husband, Mark. We're from Oxford, Ohio, and our almost 9-year-old son, Liam, has limb-girdle LMNA-C, and it's funny as I'm listening, I'm like, is that the right thing? Is that the same thing? Like, we're still trying to figure out if it's the same as or similar to what we're talking about today, but nice to meet you all.
We have a new guest here.
Hi, I'm Mark Manning. I'm from Chelmsford, Massachusetts. My daughter Madison is 4 years old, and she has an LMNA mutation.
Thank you. I think we can do this. It's not very formal. We are here to respond questions that you may have about research or concerns that you have, and so maybe I would like to start with questions from you, if you have now already some questions to ask. If not, we can start, I don't know, giving a review of what we discussed in the last meeting.
I just want to maybe ask if you could do a quick sorry. I wanted to ask the panelists if you could do a quick overview of the spectrum of LMNA disease to answer the question that the family asked in the back. Is limb-girdle LMNA disease the same as what everybody else may have? So I think it would be helpful to set the stage.
Thank you. Well, you have a...
You can start by the genetics.
The genetics?
Yeah, what they are different.
Yeah. Yeah, it's relatively complicated because there are a lot of different diseases that are induced by mutation in LMNA. So it's not only skeletal muscle disorders that have been observed, but there are also some progeria. There is also lipodystrophy and nerve system defect, so it's really complicated. And inside the striated muscle disorders, there is the congenital form, the LCMD. There is the classical form, and it's children that are affected also, but a bit older. And it's Emery-Dreifuss muscular dystrophy. And then you have the LGMD form, which involves more older people with less retraction, but yeah. You will enter in detail, I guess. And then there is also isolated cardiac disorder, so no skeletal muscle involvement. And more complicated, there are some cases where the striated muscle disorder, there are other defects that have been observed, and they are really rare cases. Often it's only one family or two families with additional symptoms, and we don't understand the reason of these additional symptoms. And mutation, they are spread all over the gene. We don't understand why certain mutation give rise to a specific disease, and some mutation or some family, in the same family you have isolated cardiac disorder and Emery-Dreifuss muscular dystrophy. So there's a lot of complexity here that we don't understand yet. And I'll let you describe maybe the phenotype.
Okay. So I'm a specialist in muscle and in pediatrics. So my knowledge about this disorder is the spectrum that starts early in life, And I would say that my feeling is not maybe the truth, but a feeling we have the clinicians when we talk between all of us, is that it's a continuum, and it is rather it's not natural to distinguish some of these difference that we call Emery-Dreifuss or limb-girdle or congenital form. And the only thing is that the phenotype starts at a different point of the life, and then there's a second characteristic is either progressive or not. And that is the things that there are things that we do not understand. Why certain patients start very early and they have a progressive course. That is the most typical way of the congenital forms. But there are also later onset patients that should not evolve very fast, and sometimes, occasionally, they may do as the very early onset forms. But usually, those who start later in terms of muscle strength, they are less progressive than the early onset forms. So there's kind of a schema, something that is more or less followed, but we have all some patients that are not the regular form. And so these things probably can be one day answered by the genetics or other researchers, because probably the lamine may be modified by other genes or even by epigenetic things like what is the activity of the child, and because we have many questions we do not understand why the face is so well preserved. For example, muscles are in the face, and the swallowing is really very preserved in even patients that are very severe, and it's very axial in the beginning, and hypotonia becomes the later retractions. So I hope that in the next years we'll be able to understand better. But from now, what we can do is try to see what is the course of a given patient, follow it very closely, and then try to anticipate things that we are thinking are going to arrive. And so, as you know, all main aspects are respiratory, are cardiac, are orthopedics, and also in lamine there's other problems like nutritional problems that are important in certain of the patients, and we do not understand very well, or why they may have hypoglycemia and probably metabolic abnormalities that we really don't understand well. But this is why it's very helpful for us to have your information, to know about things that you may think are anecdotal, but maybe when we are reviewing different cases, and to have a sense. And well, this I'm waiting for your questions. Do you want to add anything? Because Soledad has a very important experience in this disorder, and she is in the center of reference for all Argentina. And I think she, well, I'm not sure that I have more experience that she has.
But I think you have done a very good summary of the disease, so perhaps it would be interesting to hear the questions.
I want to add that I mean, there is an effort going on right now that is the natural history studies. Maybe you heard many times about these three words that is to, in this case, they are doing a retrospective study with the history of patients to detect different time points where different complications start, and that information will be very helpful to do also prospective studies of the natural history study of the disease. And I think in October, when we had the third meeting, Karsten Bonnemann, that is collaborating with France, and I think you too, right? Yeah. You know, they presented the first statistical results that they were evaluating, and there is an idea. The next meeting will be October this year, and we will have an update. I think they are trying to wrap up and publish that data, and that will be very important for the field. Without the natural history study, it's very difficult to plan a clinical trial. So this is a very important thing that is going on right now. So welcome for questions. Oh.
I was just wondering how you're getting the data for the natural history study. Is some of it coming from the CMDIR, other places internationally?
Yeah, we performed we performed a form that we thought it would be interesting, not very deep information, but we had a consensus between different clinicians and other colleagues. And so we started to put some information from the different groups. And we had more than 150 cases answered. And we wanted, in fact, we wanted to have the biggest spectrum for the childhood forms. And we put the cutoff in 5 years, onset before 5 years of age. It was just because we wanted to do like that. It could be 6 years or 4 years, but it was like that. And then there were some surprises, like for example, the new gene techniques allow to perform the diagnosis to people who do not know about the disorder. So there were, for example, in different countries that are not known to participate in these meetings, many patients that were very young. And maybe this part of group of patients are very, very interesting, because we usually detect the diagnosis when it develops some clinical features that are very easy for us to detect. But before that, sometimes parents do not have a diagnosis, because the layman AC diagnosis is not that easy to do, based initially only in muscle results or other. There's not a specific abnormality in the muscle or that helps. So usually it's like a negative results, and then there's a moment at 2-3 years of age that the phenotype may develop, and we can say, well, is this a picture, and we can bet that it is the question. So did I answer more or less?
Like for the parents who sort of, they're not, I mean, it sounds like they want to know how they can participate. Like for parents who are not under care of a physician who you collaborate with, how do they contribute, I suppose? Is that what I mean, that's frequently the question, sometimes they may not be under your group, or, so is there registry form or a natural history parent reporting form that you're generating that will try to gather those information?
In fact, to complete also the question, the registry that is doing QCMD, of course, was into it was one of the ways to get the information. And then probably, since it was necessary to have some more technical details, we needed to have results about cardiac or other subject more detailed. So probably I think the answers were filled, most of them by clinicians. So usually there are few laboratories that can do research on this gene. So when people have a diagnosis, usually they can have access to these few laboratories or people who are working in this disorder. So I think that's a positive and a negative point. I mean, if there are few people that are known to be experts, it's easier that they get information from the people that are even very far. So probably this is a positive point. Then the negative is that many people don't know that this is very rare, and so they do not try to go through this network. So probably QCMD makes a big difference, because now people may have an access to information, and I think it is it's a very good visibility for us to have a link with cure CMD. So,
yeah, and I think the natural history studies it's a retrospective one, the one that is going on, and I think the next step that will be the prospective will be the part where new families can be incorporated for that studies, I think.
yes, right.
And also, sorry, I know that there is in the planning that to study specific, to do prospective studies of, for example, the people that is taking prednisone steroids, because there are many people taking steroids, and there is not scientific evidence of their effects. So, but they are taking it, and some of them for many years. So it would be good to do a natural history study of the people on steroids.
Hi again. 2 questions. First, a thought. Thank you for joining us, all the medical professionals. Thank you for coming. You've come a long way. It's very reassuring for families to see you in the flesh and not just read about some of the work you've done. So I had 2 questions. They're not really interrelated, but one is, I'm wondering if you could talk about any insight you've gleaned from your research about maybe issues of progression. It seems curious that there's such a spectrum of progression from early onset as compared to a late onset where it's the same disease, but it's such a slower pace. I'm wondering if there's any analogs even of similar kind of things in the medical community, but specifically if you've gleaned anything in your research about this rate of progression, why it differs. And then also more kind of hitting home is, I'm wondering what we can do for you in terms of, is this a funding issue? Is it just information that you need? What keeps this going and gives you the steam you need to keep going?
You can try to
For the
tell us about your mice, the mice. How do you see this?
No, but
or maybe also it's helpful a little bit of the genetics, or because there are different mutations in the same gene, and that
yeah, no.
Or you go wherever you.
I don't know about the progression, what makes it so different, and we don't understand also why the onset is so different. So there's really a lot of work to do there, but
you can talk about the consequences that you have found in your studies that may explain a little bit why the disorder may happen, like the differences in lamine, for example, for progeria is different. The consequences that is making.
Okay, so behind the skeletal muscle disorders. So for the other disorders, the ones that are not involving the muscle, we know that there are specific mutations. They are known. It's like for the progeria, the LMNA gene encodes different isoforms, and one isoform is pre-lamine A, and it has to go to maturation to give the lamine A. And in the progeria, there's a mutation that inhibits the normal maturation of prelamine, and then you not having the lamine and having this prelamine form mutated is what is giving the pathology. For the lipodystrophy, there are also like more than 80% of the patients get the same mutation, and it's a mutation in a specific domain, and it has been shown that this domain is involved in interaction with other protein, and this domain is involved in the interaction, and the mutation inhibits this interaction, and it's then the pathway or the signaling after this interaction is not occurring, and that's what leads to the pathology.
So the protein that is supposed to form with these different mutations provokes that this protein is formed, but in different ways. Some proteins will lack the last part, the end part, or some others the initiation part, or or cut in the middle, stop. There is a stop signal, and there is just instead of 300 amino acids protein, it will be 150 only. So these proteins still, some of them are a little bit functional. So that's part of the spectrum depending on the mutation. Am I right? And
yes, that's more or less the case, yeah.
Okay.
So what is makes the thing more complicated is the straightened muscle disorder, because, as I said, mutations are found all over the gene. It's all kind of mutation, so it can be a missense, so just one amino acid difference. It can be a stop mutation. It can be mutation that will lead to a premature stop mutation. So it can be all type of mutation everywhere on the gene. And now with the knowledge that we have on the protein function for the straightened muscle part, so why this gene that is expressed all over the body just affect the muscle and the heart. And we are thinking that lamins are important for the nucleus and for the cell to sustain the stretch, the forces that from the environment, and muscle and heart are under constant stretch. And if you don't have a normal structure of lamin, you cannot sustain this stretch. And then the cardiac cells go while they are dying, and the muscle cells, it's a bit different because it's a fusion of a lot of cells. So, but you are also damaging the muscle with that's an hypothesis. But and the specific things about lamin is that the protein, they form a network. And what we think for the straightened muscle mutation is that we are affecting the formation of the network. And we think that for the congenital form, the network is not forming at all. So we are working on that, but I don't know about the progression. But for the different disorders, that's where we...
Can you explain that network a little bit more?
Yeah, so if you visualize a nucleus, below the membrane that forms the nucleus, there is a kind of network. It's a cage formed by the lamin. And this cage has many functions, and one of the functions is the resistance to this environmental cruise of stretch. It also regulates how the DNA will be used, what will be expressed or not expressed. So there is really a tones of function of lamins, but
let me go a step back. You most probably heard that the mutation in the LMNA is in a protein that is part of the nucleus of the cell. So we have the nucleus of the cell where all the DNA is inside, and all this transcription of message that will go outside the nucleus, will go to the cytoplasm of the cell to produce the protein there, and then in the case in the CMDs we have the extracellular part of the muscle, that collagen 6 and llama 2, the mutations are in proteins that are there, structural there. Then we have some that are inside the cell, and this one is the only one that is inside the nucleus, and that mesh that it forms is to maintain the, it supposedly maintains the structure of the nucleus. And when that mesh is not there, the nucleus is floppy, and all this stretch in the cardiac or the it gets stressed and ruptures.
I just wanted, I think it would be helpful to have a couple of basic definitions, because not everyone in the room is yet educated in this field. So LMNA is the name of the gene. The gene codes for a protein called LMNA-C, and that protein forms this mesh or this structure that holds the nucleus, kind of keeps its shape. The diseases that, there are many different points along that gene that can be mutated, and that's why there's such clinical variability in the way it manifests. So mutations at some point within that gene cause deformed lamin protein, and that then causes a whole host of diseases such as the congenital LCMD, or the limb-girdle muscular dystrophy, or the Emery-Dreifuss type that is typically later onset in adults, or this progeria, or an isolated heart muscle dysfunction. So there's a whole spectrum of clinical disease. It's all caused by mutations in the same gene, but they're not the same mutation. They affect different parts of the gene, which then alter the way that protein is formed and the way that protein works. So.
What's your son have?
So LMNA is the gene. Yeah. Lamin is the protein, and the diseases are, yeah. So I hope that's helpful. Yeah. Okay.
I have a question along this line. So we have a couple different families here who have LCMD, who, as far as I understand it, have different mutations that are causing the same disease. So then, is there any explanation for why that is?
You guys want to take that one? I think that there's a tremendous amount of genetic variability. We use the terms genotype to describe the gene defect, and then we use the term phenotype to describe clinically what it looks like. How does the patient look, what symptoms, what signs do they have? And so there's tons of overlap. And so you can have two patients that look like they have the same type of muscle weakness, and when you identify, there are patients with Emery-Dreifuss muscular dystrophy, in fact. Most of them have gene defects in the LMNA gene, but some of them have defect in a completely different gene causing completely different protein. So there is not this idea of one gene, one disease. So there's lots of overlap. So I hope that that's helpful.
The genetic background, because we are talking about only one gene, and we have 30,000 genes according to the human genome. And so all these other genes that they have different forms, they can modify also the course of the disease. And that's one area of research that is going on in the whole world, but to find modifiers, because this is not only happening in this kind of diseases, that there are some modifiers. You have heard, we can go to all the other end in Alzheimer's disease, and the Alzheimer's that we know that is genetically provoked. Also, they have a different course of the disease, and there are some other things for sure that modify. That's difficult to fish to find. But.
Okay, so you have stories. I have also a story, and so when we first identified the first patient with these mutations, as I said before, I had another number of patients that resembled, so I thought they could have the same disorder, and then I sent it to the genetic lab, and I sent 3-4 DNAs, and I said, I know what it is, and they said, well, let's see. And the story that I want to tell you is that one of the mutations of I think the first patient was the same mutation exactly of one of the others. So this is not by chance. It's de novo mutation is something that arrives for the first time in the baby. So this means that probability is impossible that this happens again, like just by chance. So this means that certain mutations are really like giving progeria, but giving a severe onset or an early onset. And even there are many heterogeneous factors that we don't understand well. At the end, most of the patients who have the very early onset phenotype picture, they share one of a number of mutations that are known to have more more pathogenicity, or that are more prone to become something that is progressive or has a different course than the others. So this is the story that I have, and I think that can be improved by the scientists because and when they saw that there were several mutations that were very frequent in the more severe or early cases, they tried to build concevoir to perform a mouse model with one of these mutations, and it ended to be the most severe model. So there's something that it may be explained by genetics or some of the studies that Anne is taking now at this time. So, but the only difficulty is that heart and muscle maybe is not the same in terms of the mutation may not have the same consequences in heart and in muscle. But also the children who are more mobile may have been using the heart more than the others. So you can never know if it is the mutation or is the use of a given muscle. As I said, why face muscles and tongue and swallowing muscles are that different than the others in other parts? Why proximal upper extremity muscles are very wasted and they are not wasted in proximal lower limbs? So this is difficult, but for sure there's something that explains it, because it's a very systematic, it's a pattern. So when there is a pattern in clinics, we try to find a solution where we discuss with the scientists and they try to see if this is the same in their models, and sometimes we can have a clue to move forward. And then to answer to the question how we can move is by making this kind of of meetings. Every time that we meet with you or each other with a common idea, we are having, I don't know, several hours or days focused, and all the things that we are thinking during the year, maybe we have the opportunity to ask or to share. And sometimes it's some anecdotal thing that allows to move, make a step. So we are trying to do the group of experts that are interested, these different meetings as we can, because there are a lot of things to do. And I would say the bottleneck is mainly the time and the number of patients, of people who are really motivated and that know about, because many people could be very motivated to join us, but they don't know what it is. So you need a part of training into the knowledge in pediatrics, in neuropediatrics, in neuromuscular, and or in cardiology. So this is a very specific point. So I think the bottlenecks are those, and concerning the time, is really fundings, of course, but you need also the people and the questions. You need to have questions to try to answer them. And I think the most important thing is to try to see what is not known, a prioritization of questions. And it's difficult to tell you how we advance, but when we find each other, we can think that something is interesting, a question, and they say, well, we will think how we can answer this question. So it's step by step, but it's a question of community.
And this is, I mean, this is the pathway. They discover the mutation in the clinics. They see the patients, and then the scientists they create the animal model with the same mutation to be able to study the disease, because we cannot do it on patients. So and to complete the loop, they are doing gene therapy on that animal model that they can be translated. She's the one that is doing the gene therapy now for specifically for one of the mutations in the LMNA.
I feel like I have so many questions. Hopefully I don't overwhelm you guys. Did you say there's 150 cases of?
Yes, more than that, but at least we had answers from all over the world, China, Australia. So you maybe more 200 for the registry, for the retrospective registry.
Yeah, something like 200.
200, yeah. But for many, we do not have a lot of information. So
that's for the natural history.
For the natural history.
So is that
Not total number of patients.
That's all LCMD?
No.
Folks?
Oh, it's
or just any LMNA?
No, until 5 years of age. We asked, I would say that how much, 20%? 20% of these patients were...
So 150 folks that you know of in the world that have LCMD?
The very early onset was 20% maybe of this group, but yeah. Of 200. So maybe most of the patients have not an LCMD, or what we describe as that. There's not this early normal course or retarded acquisition of milestone, and then a progression with head drop. These are the typical LCMD, but I would say 20% of the patients that were in this registry or collection of registries were really LCMD. So then many of the patients have started maybe in early childhood, but they do not have this head drop, and they start really with the axial rigid spine, and then they develop more the Emery-Dreifuss phenotype with the years. This means that they start to have elbow contractures, and they are ambulatory. They then may progress to a severe form, losing walking, or they stay in this retractile phenotype. So most of them are this group in the middle. They are not the early, very early, or the typical Emery-Dreifuss more late onset. Yeah, all LMNA, yeah.
I don't think that many folks are on the Facebook community, right?
No, yeah.
I don't know, I think it'd be helpful if whoever is capable of doing that to get all of those people together on that community so all the families are united. I mean, that's what started the Duchenne and Becker movements, right? So the more we know about each other, the more we can help you guys and help us. Yeah. There's a translation button, isn't there? Maybe that would help.
Yeah, but you may be interested in sharing, but there are families that have a physician, they have the answers, and they don't look for other things, other that don't want to know more. It depends on families. It's true that I think it would be useful, and I try to engage them in meetings or in having more information. But that's not the case for every family. In France, there are not even organized in a number of. For example, in Spain, I come from Spain, so one of the patients I diagnosed created a foundation who is trying to move a lot of things. And when I talked to them, I said, well, you have to do two things. One is to try to improve the quality of life and treatment of your child, and trying to do the same for the other parents. But you have to try to make us move forward with funding and everything. If you can do both things, it's perfect. So he started. He moved it, and it is amazing. Yeah, and it's amazing. I cannot imagine that this family. I received an email sometime, and they said, "Oh, we are doing." It is amazing how people can move, and I would have never done this. And they are paying for researchers in Giselle labs. So it's incredible, because in France, for example, is AFM. Exists, and it's a big, huge association. And for me, it was strange to know that this foundation is paying for research. But I think maybe that's the point. We have to know what we want to do, and then people arrive. It's a natural thing.
Funding is on my mind a lot, because obviously the number one goal is the cure, and there's not many of us, which probably leads to a lack of motivation for private equity, for universities. So one of the things I've thought about is I've read about all the other diseases that have LMNA mutations, like progeria, and I think there's even a neuroblastoma cases where it's associated with an LMNA mutation, and then I think the director at NIH is researching it for the fountain of youth, anti-aging. So do you see it being possible that, instead of just banding with congenital dystrophies, that we, the few in this room, are also, bonding with families that have any kind of LMNA mutation? Because it sounds like any LMNA mutation makes sort of the cell fall apart, the nucleus, the envelope. It's the lamin protein, right? And so if you get to the bottom of that answer scientifically, doesn't it help all of them? And isn't that our ticket potentially for funding? I don't know if you guys have any thought. Like, is Giselle's lab the only lab besides the Institute of Myology that's researching You know this and the others?
No, there are many others.
Sorry, it's like 5 questions at once.
No, Giselle's lab is not the only one working on lamine and straightened muscles. There is Howard Warman in New York, that is also working on that disorder. And there are teams that are not only focusing on LMNA, but they have some program that are specifically addressing this question. And there are teams in Spain. There are teams in Italy. There are. I think that European part is really strong. For the United States, there is Anne Bank that has also a program to look for drug for the disorder. Our team is really focused 100% on lamine, but there are a lot of other teams that have some specific programs for this.
Are you guys also funded by like the Progeria Foundation?
No.
Could you be? Because you're focusing on what's important to them. I mean, couldn't that help funding?
Well, right now there is one program in our group that is working with a team in Marseille that is specialist of the progeria, because I think it's a bit difficult to work together because the questions are really different. The organs that are affected are different, and the reason why they are affected is different. So the answer that we will have will be completely different. And what we understood from progeria is not helpful for congenital form. It's helpful to understand more of its function, but it doesn't serve immediately for congenital, and vice versa. It's really completely different.
The toxic
yeah.
Toxic.
Sorry? The physiopathology is completely different, and the what is altered in the function of the protein is completely different. So,
what is causing it?
Yeah.
So the mutated lamine is different. That's not in both cases.
Yeah.
In progeria, it's more a toxic prelamine that is probably. They try to
So it's for the progeria, we know that when patients are aging naturally, they accumulate prelamine, and among patients that have progeria, we know that the more they have prelamine, the more disease they are. So it's really due to this, and there is nothing comparable in LCMD. There is no accumulation of prelamine on everything, and we don't have this toxic effect of accumulation of prelamine in congenital form.
Yeah.
So.
And in the congenital form, what you see is that they have somehow a difficulty to develop fibers. We see small fibers in the muscle when we see the muscle biopsies, and then sometimes it becomes a degeneration, like we call dystrophy, a dystrophic muscle, and somehow the processes that we see as clinicians, and when we talk and we see the muscle specimens, is that this disorder, for some certain reason, at a given moment, the muscle is at a given point doing the work, but then he starts to not to be able. So that we do not know why this starts to happen.
So I was just wondering if you could tell us a little bit more about your study, about your mice, what you're looking for, and what you are hoping to do in the future.
So we developed in a lab. We have 2 mouse models: one for the classic amyotrophic muscular dystrophy disease, and one for congenital form. So we reproduce in mice one of the mutations that has been found in LCMD, and if we compare the 2 mouse models, we can really clearly see that one is really reproducing what happens in amyotrophic muscular dystrophy, and the other one, the congenital mouse model, is really severe. The mice are, the difference compared to human is that we need to have 2 copies of the mutation to get the disease, and it's the same for every kind of disorder that you want to reproduce in mice. In patient, 1 copy is sufficient, but you need 2 copies in mice to have the disorder. And the mice that have the 2 copies of the mutation, they are dying in 2 weeks. And we see that there is muscle defect. The cardiac function is not that much altered at that time. And so apparently that is a bit different in patient. And we also saw this metabolic disorder that we don't understand yet, but there is a problem of development of the adipose tissue, and so we are not actually working on that metabolic part. Now what we are trying to understand is what makes LCMD mutation different from EDMD mutation. And as I was mentioning earlier, with the use also of patient cells and our model, we saw that the lamine that is mutated is not accumulating in the network underneath the nuclear membrane, and it's fine throughout the nucleus. So we think that the defect of formation of the network is one of the reasons why the LCMD mutation is so severe. So we're trying to understand why they cannot form this network.
So you think it's more structural?
Yeah, I think it's structural. Yeah.
Consequence.
Mm.
Okay.
And then if we understand why they don't form this network, we may try to intervene on specific aspects that might change the behavior of the protein to force them to do this network. But we are not there yet. And then we use cells from patient and from the mice to try to design therapy. So the gene therapy has 2 aims. The first one is to reduce the mutant protein. So we use small RNA that will specifically recognize the mutant RNA. So the mRNA is between the gene and the protein. The gene is transcribed in mRNA, and then the mRNA goes in cytoplasm to form the protein, to encode for the protein. So we are targeting this specific mRNA to destroy it, and we just stay with a wild-type mRNA. So to be able to have only wild-type protein that is formed. But what we know from the patient is that having only 50% of the protein level gives a disease. It's isolated cardiac disorder. So it's adulthood, around 20 and 40 years old patient, they develop this cardiac disorder. So it's still better than the congenital form, but we also have to address this decreased protein level, and then we want to come with a wild-type version of the gene to recover the normal protein level.
Okay, so they are doing both ways. One, to reduce to inhibit the bad protein and leave the. I think we have to go back that the mutation in this disease is a dominant mutation. So you have 2 copies, one from father, one from mother. Although in this case it's a de novo mutation, so it appears, but it will appear in one of the genes. The other one will be okay, but with only one mutation is enough to express the pathology. So they are trying to inhibit that bad gene and let the other one alone, that is a good gene to produce, but probably it's not enough to overcome the problems because 50% of the protein from the good gene maybe is not enough to form the good mesh. So they are trying to also introduce another gene, the good gene. So they will have the normal gene, the bad gene that they inhibit, and they add the 3rd copy that is good. So we will have one of the copies that you didn't touch, one of the copies that is bad that you are inhibiting, and then you are adding a new copy. So to have more of that, to compensate and have more good lamine AC to form again that mesh. I don't know if.
So she was asking if we were using the CRISPR-Cas9. So it's a new technology that, it's kind of scissors where you cut a specific part of the DNA and then you replace by a good part. So here it's really the genome that we are touching. It's not the mRNA. There is a team in Spain that is trying to address these questions through CRISPR-Cas9 technology. We didn't do that because, there are two reasons. The first one, I would like to, if possible, I do not want to touch a genome. I think it's, I don't know what we will change besides what we aim to change if we touch a genome. CRISPR-Cas9 is really difficult to work to change a mutation to, this specific kind of events to be changed. It's really difficult. It's easy to create a mutation, but you don't know which kind of mutation you will get. So you can touch a specific part. You can address a mutation in a specific part of the DNA. You can really choose that, but you don't really know what change will happen. You cannot control that in a good fashion. So to change a mutant back to a normal copy, it's not that easy. And the second reason why I didn't want to go through that path is also there is a recent publication that shows that beside the part of the DNA that you want to target, there is a lot of off targets, and we don't know what that can do in the body. Maybe you are worsening the patient. So I think there's still a lot of work to be done to for the efficiency and the efficacy of the CRISPR-Cas9 before we can really use it to cure mutation. And
what's the timeline like on the therapy part? You're doing your mouse models now, but just curious, what timeline you're at this point now? So what going forward? Is it months, years? Is it all dependent on what you see? How long before, obviously, you start involving people? Is that reasonable? What are some of the next steps? To speak to that a little bit.
Hard question.
Yeah, I'm sure.
Yeah, obviously everything is depend on the result that we will get. If it's a positive result that we'll get in our mice model, I think we need at least 2 more years to see if it's working in mice. And if it is working in mice, then we can try to set up a trial in patient. But I have no idea how long it takes to go there. I'm not really skilled in that.
And this is going back to funding again, because this project is funded by Andrés Marcio Foundation and Cure CMD, and the grant will end soon. In this case, we have MD UK that they have some families there with LMNA, and they want to collaborate with the funding. We were talking with Jenny. We are very happy that she stepped in she came to me to say, okay, you have some projects in LMNA that needs fund, and maybe we can help there. And so all these things are happening at the same time, and funding and sustained funding is crucial. Because the timeline can be, as she said, in 2 years, you know if that specific gene therapy works or not, and then you have to make the other plan that is to translate that. But when you have positive results, you can show other people, try to engage companies and make it scalable.
Is at that time that you have to think about the merit and all this. So I think that maybe at this point there is not a real treatment specific of the disorder. So the people are doing their groups because they recognize themselves, like pediatric or Emery-Dreifuss, and they don't mix now, because in Spain, for example, probably the Emery-Dreifuss patients are on the side, and the congenital forms are in the CMD group, and in France it's similar. But when there's a specific thing that is for the gene, then at that point, if all of them share the muscle involvement, the heart involvement, that will be a moment that we have to emerge all together. So it will be more easier.
And one thing that I would like to add is this approach is goes for, as you said, every kind of LMNA patient, not necessarily the congenital. It can be for everybody. But before going to trial, we really need this natural history of patients. So that's also something that we are working on, but it can also take years. And we really need to know how the patients are progressing in the disease. To find what will be measured by the clinician after we start the treatment. If we don't have the clear way to evaluate the patient progression or this, we cannot go for a trial if we don't have really precise measure to go on.
So the question now is, we would like you to talk about your children and to explain how it evolved, so we have an idea of what is the population here, and maybe we can move forward with some questions that you may have in terms of the doing better, because we believe that the parents may be researchers themselves, and we are not many. So I think you know very well, we know very well. I think we can make a team. So that is something that we will appreciate.
Yes, and also because we know that these therapies are going to take time, we cannot forget about the other issues that are life-threatening or cardiac involvement, things like that. So we have also to study those problems and come up with some solutions for that. So we have to diversify the funding. So I mean, it's a complicated, but I mean, this is very necessary and important, and from here it will come up some.
Hi, I have a question. I have a 10-year-old son who is the affected member of our family, and we've had his heart tested, as you say, through all the grams every 6 months. And can you highlight a little more about the 10 to 14-year-old you just mentioned about cardiac events? That obviously struck home with me.
We need more information about your child to answer, because I need to know where in this spectrum your child is. We need to know how he evolved when he started and how is it going, and so we can put your child in a kind of pathway. So I leave you.
Do you want to make some comments?
Absolutely, sure.
Oh, okay. Yeah,
is that a better session?
Yeah,
okay.
Yeah, that's true. Yeah. Thank you. Yeah, no. Thank you. Yes, right.
I was just going to suggest that, to speak to your point, that the families just go around and talk a little bit about their child who's affected, and more of the phenotype that they've seen, and also maybe any medications or treatments that they're currently on, just so that you can get an idea of what we're doing as families. So Charlie is going to be 9 in September. He has been in a wheelchair since he was 2. He's never been able to walk. He does not have the dropped head. He does not have any cardiac issues yet, although we are under Dr. Heller's care at Connecticut Children's, and he does have the Link monitor implanted, so not part of the study, but he does have the monitor implanted, and he is currently taking prednisolone.
And beta blocker.
And a beta blocker.
From what age?
About 18 months, 2 years, we started to see, 15 to 18 months, we started say something's up, and the diagnosis was 2.
Okay. And the prednisone?
Oh, prednisone.
Since he was about 3 or 4.
Okay.
Yep.
What would you say after the treatment started? What did you do?
We initially didn't have any complications. We did notice that his strength kind of maintained to the point that it was at. It never really got better or worse, but he didn't degress, he didn't progress. So we've kind of kept it steady at the same dose.
What about the contractures?
He does have pretty severe contractures in his ankles, his knees, his hips, his elbows. Mostly his hips are the most affected.
Okay.
Yep.
Concerning the spine?
His spine is pretty normal to rigid. I wouldn't say it's rigid, but it's fairly normal. So he's a little bit different in that aspect because he doesn't have the dropped head. He doesn't have any like spinal issues at all, scoliosis or anything.
Okay. The respiratory?
We haven't had any issues with.
No problem.
Nope.
Okay.
No pulmonary issues.
And do you know the mutation?
No.
No.
We don't know.
Go this way.
Christian is 16 months, and we noticed the doctor first told us that he had torticollis because he was like tilted and weak, and most doctors told me that it's probably no big deal, and I insisted. We went to a neurologist, and one thing I noticed early on is when he was itsy bitsy, when we first got home, I kept hearing this like banging in the bassinet at night. I guess he was trying to lift his arms, but they kept falling because of weakness. Another thing I noticed was he wasn't tracking his eyes. A lot of the doctors said that. He obviously had no reflexes on a lot of our appointments. He's got, a dropped head. He never was able to sit. He's really, really happy, really, really smart. Is there anything else? We're not on any medications at all, but we clearly need to research prednisone with our team.
Madison is about 4.5, and we started to notice something about 12 to 15 months is really, her ankles contracted first. We brought her to an ortho thinking we'd get some braces and be on our way, and that was the beginning of, at that point it was just her ankles, and then by the time we got to the neurologist and he asked her to lift her arms, she was unable to, and we were both surprised at the moment that she couldn't do that. She doesn't have a head tilt. Just recently it's a little more forward than normal. She is on prednisone, has been for 2 years, about 2 years. And I think we noticed that it gives her maybe just a little more, I wouldn't say more muscle strength, but just a little.
When she's not on it.
Yeah.
A loss of function.
No, she does not walk. She can't stand.
Yes, we do.
We do. Yeah.
I know I need to do it. It's a big deal.
Okay. Another family.
Did you say Madison never ambulated, never stood?
No. She did sort of army crawl for a little while. She did stand, I'm sorry. She did stand holding things. I have a picture, and I didn't know that was going to be the end of it.
Picture in your mind too of that.
Yeah, but never really. She did sort of army crawl. Yeah, it was. Yeah. It was about 15 months. And then within 6 months, within 15 months to 2 years, it was very clear.
Does she have, if you don't mind me asking,
No,
does she
please.
have, is there more strength kind of the top of the leg?
Oh, definitely. The top. She scoots on her bottom all over the room, but she can't lift her hand to her mouth. She can a little bit. She'll put her head down to do that.
Her elbows on a surface to be
Yeah.
able to assist her.
Yeah. Yes. And that's one thing we want to talk about because she's braced all the time and we don't feel good about it. I don't know what the right thing to do is. So that we noticed. She saw Levi and said, he has naked feet. And I was like, I know. I'm going to talk to Levi's parents about that. So that's.
I think another thing unique to her is she inherited the mutation from me. I've since been diagnosed with laminopathy. But my mutation has also been confirmed to be mosaic in nature. So obviously I'm not affected with a neuromuscular condition, and that kind of confused every doctor upon my genetic results.
Was that as a result of Madison's diagnosis? They tested you guys?
Yes, and I came up positive for the same mutation.
That sounds like the Olympian and the. Did you read that article? Yeah. Have you guys heard about that article? There's two women. One's an Olympian and one is affected by, I don't know if it's LMNA or Emory.
It is LMNA.
So she did research, she found the Olympian, and the Olympian has the same mutation, but instead her body just creates gigantic muscles, strength. And so listening to him and thinking about her, hopefully that leads you guys somewhere.
Do you know about them? You have to look them up. It's a very intriguing story. A few years ago. She also has partial lipodystrophy. She has unrejected LMNA form of partial lipodystrophy. So she's very into the science aspect of trying to figure out what's causing this. But she was able to find this other woman.
They hired her at a university too to do some research. I forget which one. The woman who has it.
She's working with someone at the Boston Children. I know she's working with the group in Boston Children
So
or now to try to figure out.
Yeah, she's in the Olympics. Like I think she's track.
Olympic. Huge.
Yes,
Okay.
she's huge muscles, very strong. And then there's another woman who has a mutation, and they've gotten together somehow. I forget how she did it, but they found out that the person who's very strong and healthy, her body just did something different with the mutation somehow. Sounds like similar to him, or some way there's no.
I think she was looking at the picture of the, I think the shoulder girdle.
Yeah, and her.
I think she has some
Because her
atrophy
butt
of shoulder
muscles too,
girdle, but then
they
there's certain patterns.
Yeah.
Jill, Dopfleles, and there's a ton of articles online. So far, that's a very interesting story. The Olympian is Priscilla Lopez.
She will be someone for like a good modified gene, you know,
Yes.
say what is
Yep,
modified.
they've been studying
so that
their
it's different.
genes to figure out why it's causing her to be more weak with the LMNA form and Priscilla Olympian to have the opposite effect on her muscles. But it's the same, I believe it's the same mutation.
One other thing I didn't say about Madison that I should have, she does have cardiac arrhythmias. She has the link monitor. We are part of the research study in Spain, and we've had several reports of arrhythmias and things.
And what about contractures?
Yes, her ankles and her hips and her knee, one knee is much worse than the other.
I don't want at all to cut this conversation, but it's 12. I think the lunch will be served soon.
Yeah, can we finish the family?
Oh yeah, of course. No.
Okay.
I'm just telling
Just,
that
yep.
we cannot go
Sure.
2 hours more.
So our son is Levi. We noticed at 2 months he was just a little floppier than our other kids had been. So around 6 months, he sat up one time, like Kyle sat up on stage, and that was the only time. He was diagnosed just after his first birthday. He was diagnosed with muscular dystrophy just before, and then Dr. Seljan at the Mayo Clinic diagnosed him with genetic testing as LCMD. He has severe foot contractures. He has no use of his arms. He can't lift his hands. He uses his feet to do things, so we stopped bracing. He literally lays on the floor, and with one leg, he'll brace his other foot to use the iPad with his toes. It's amazing. You have to... Yeah, he also pinches. It hurts. And he tries to pull people's pants down. That's his new thing. Yeah, his head drop, he has no control of his head. So he has supports underneath his chin that keep his head up. Without that, I mean, there's no head control at all. Our current issue is vomiting. He's G-tube fed. We have a GJ. The J, for whatever reason, does not work for him. He vomits regardless. No formula, obviously, because it's in the J. But that's our current issue. We would love to start prednisone, but our neurologist wants us to get the vomiting under control first, obviously. So. He has a BiPAP at night. Yep. Yep. So I would say knee contractures, hip contractures. His spine is good so far. So heart is checked out. We have that checked out every 6 months, and so far so good on that. He is cute as a button, yeah. You should note that. We have considered it. We've talked to his cardiologist, and they said once.
No.
Okay.
I'd like to speak, yeah. Like I said, our son Liam is almost 9. He has limb-girdle, LMNA-C. He hit all his milestones, walked. He didn't crawl. He kind of did the Buddha scoot, we called it, like the getting across the ground. He now has a bit of the dropped head. And his elbows are very tight. His spine is very tight. His ankles are tightening now. He has the winged shoulder blades. He's very high functioning. He uses a wheelchair for long distances, and here I'm noticing he's using it a little bit more than like at school because he's looking around at all the cool wheels. He does very well. He's very high functioning, super bright. Just gets very tired, but what else? His heart looks really good. His PFTs are actually like, they say, I'm sorry? He's almost 9. His PFTs are actually, they said, like better than normal kids. Like he's got like really, he does walk. Yeah, he can walk, he can run, he can barely, like he can get a little jump. Try to keep up with his sister. Yeah, he's on nothing. Like we give him melatonin just so the kid sleeps. Like,
What age was this discovered? It hits all the milestones.
I'm sorry.
He hit all the milestones.
Yeah, he hit all his milestones. I'm an early childhood professional, so I spotted it a little earlier. Like Dr. Jim Collins is our neurologist, and he said normally people wouldn't bring him in at this point because it's not noticeable. But he was doing just little things. I was like, that's a little off.
I'm a little anal retentive, so I actually wrote down the mutation because that's just how I am. So Alec is our son. He's 10, and he here help. Okay, sorry. I like standing. So he's 10. He was diagnosed at 1. I think the date is 5 of 2010, and his birth date was 2 of 2007. So that was a 6-month process after we had already gone through the initial diagnoses. Neither one of us had a mutation, and he has the dropped head. He has contractures in his wrists, like you said up front. His muscles here are his abductors are better than his inductors, or vice versa. The outside muscles are better. He can kick. He stood once. We have the one picture, as everyone else does, and he never. We actually had a really awesome in-home child provider who diagnosed him, who came to me and said something's not normal.
Before his first birthday.
Before his first birthday, he's floppy when I pick him up, and so that's how we sort of had the first.
We were blaming it on we both have big heads.
Yeah.
And he was just having
Yes.
trouble supporting.
Yeah, because our first kid also,
At 1 year, she said, no, there's something wrong. And within months, we knew. They did a muscle biopsy.
So what else? Oh, he's on a BiPAP. He did a sleep study. And we also live at altitude in Colorado, so we have he's on oxygen as well, and breathing support. He sleeps a ton. And he's on prednisone. How much prednisone?
Just under 2 milliliters.
Just under.
And we battled with one doctor to get him on it to do something. I don't remember what it was 5 years ago, and fought to get him off and have to go through that process. And then within a week, we were back on it from another doctor, and we've reduced that volume in half here about a year ago, and it had, I think, an emotional impact on him.
He was a real pain in the ass for a long time. Because of that, he's, just obstinate and difficult and would have bursts of emotion, so we've toned that down a little. But we definitely noticed more strength because of the prednisone. And we have the GI issues. He's got a G-tube and a J-tube. Oh. I actually thought we were the only ones who had 2 instead of 1. But he's fed through G-tube.
J. We feed him through his J.
I'm sorry. Feed him through his J, put medicine through his G-tube. And he's on a ton of medication, ciproheptadine,
Miralax.
Mostly for GI, just
for GI issues because he's
it took us
vomiting.
years to deal with his GI issues.
Yeah.
And then we finally, Miralax, miracle drug. I mean, it changed his life, and now he's not sick half the year, just maybe 2 months of the year.
Yeah, in first grade he missed 40 days of school before Christmas. So, and he's in regular school. He has a para assistant, and we're starting to, as you guys always know, everything changes. But we kind of got the GI issues in line, and then they come back once every month, maybe once every 2 months. He'll have a 3 or 4 day bout, and we see the muscle clinic at the Children's Hospital in Denver. Rock on, they're awesome. What else? What other? We talked about the BiPAP.
Very fused spine, very rigid.
Oh, yeah, rigid spine. He's never, other than the one picture, he's never walked. He didn't scoot. He didn't crawl.
He scooted a little.
Little bit.
He didn't crawl. He rolled.
Yeah, and he can still move kind of from one side of the bed to the other a little bit. He's in a wheelchair full time, of course. But yeah, that's super smart. Super bright. None yet. Yeah, I mean, we get it
once a year, and they haven't noticed anything issue-wise. We don't have anything.
Nothing.
We just take them in once a year.
No, we do not. We were just talking about that. There was one point where we did it was like a 2-week test of some kind that they went through, and since then everything was fine. But that was a couple of years ago.
Okay.
There you go.
Okay, I think we have to wrap up. I don't know if someone has a specific question that needs to be done now, but we are going to have another LMNA meeting organized by Andres Marcio Foundation and ourselves in October. I promise to. I'm going there. So I'm.
It's in my city.
In your city. I'm promised to give an update after that, the webinar update about that meeting. And I'm also reachable by email to explain some of the biological things, because I'm not a physician. But I mean, your questions are always welcome. I cannot promise to reply very soon, but I will have it in my mind and do it.
What I wanted to say is that before leaving, you have shown the spectrum of the disorder in all your children. So you have from very, very early non-walkers to elders. So you should maybe make a group yourself of natural history. And you have patients who are on corticosteroids, others who not. So maybe if you can give us your retrospective story and the mutation and the perspective from today with things that maybe we can give you the form. So somebody, your doctors or even you can answer probably for.
I think tomorrow
we can't think about that, okay?
You will be tomorrow in
the
meeting, right? Tomorrow is more specifically clinical. Finally, we derive to that, but this was more research focused.
Because you know, for example, you have a person in the state that is very concerned about the cardiac, and then other information you can send in form. So it's the most difficult. The cardiological technical studies are more difficult than others. The others can be in just regular reports. But maybe we can do something about. Thank you very much.